Today we have one more young and dynamic professor, Sir M.Y. Srinivas from Microbiology Department. Please mute your
Joy Brotnath.
Yeah, video, voice. You just mute your phone. Okay. So young and dynamic professor M.Y. Srinivas, he is presently working as chairman. Just 2 days back only he has occupied the chair in Department of Microbiology. So he is again completed his MSc and PhD from University of Mysore. He is alumni of our university and very active and dynamic researcher as well as academician. Yeah, he did his postdoctoral degree in United States of America, and his field of interest and specializations are microbial diversity and prospecting, mycology, mycotoxicology, food microbiology, and probiotics. He has obtained in his name Indian patent in 2021 for the work entitled Composition and Method for Biodegradation of Chemicals and Application. Sir has obtained a lot of grants, more than 1 crore from various research funding agencies such as Science and Engineering Research Board, Government of India, Kennesaw State University, USA, UGC-DST, KSCST, Indian Institute of Science, Bangalore, and he has deposited Levulactobacillus species genome sequence in Gene Bank and published more than 81 research papers. Almost all the research papers have published in reputed foreign journals. He has contributed more than 16 book articles, guided 6 PhD students, and more than 44 MSc students for their major project work. Currently, about 7 students are working for their PhD degree. He has presented so many papers in the national and international conferences, and his H index is 22, I-10 index is 40, and total citation is more than 1,300. Of course, with all these credentials, he is a member in various academic bodies of both University of Mysore and other universities. He has been awarded Shastri Indo-Canada Fellowship in 2017. In this context, he has been visited to Ottawa University, Canada. And also he has been awarded Raman Postdoctoral Fellowship, Arkansas University, USA in 2014. International Faculty Development Award also he has obtained in 2010 and 11 from USA and India. And one of the another interesting prestigious award is Young Scientist Award from Department of Science and Technology in 2010, New Delhi. He is, of course, associate editor in Frontiers in Microbiology and other journals. With this brief profile, I would like to welcome once again Professor Srinivas, and over to Srinivas. Thank you.
Thank you. Thank you very much, sir.
Thank you, sir.
Yeah.
Okay, sir. Share.
A very good morning to all of you. I'm very happy to be here. Like previously, I was sitting like you, and today I got an opportunity. Like a couple of years, I am getting an opportunity to talk my own family members. And I was told by Basurajappa sir that there are 23 participants in this refresher course. And in that, some of them are microbiology, some of them are biotechnology, and many other life science fraternity. And he also said that it is a heterogeneous group, where very active group. And in the previous discussion session, you have shown a lot of interest, and also you have interacted a lot with the resource persons, and really that is appreciable, and which even I like to have an interactive session rather than teaching. The biggest drawback of this is I feel like I am speaking to computer actually. I don't see the interactive faces, either the active faces, and it's very difficult to speak simply for 1 hour looking into the computer. The moment when Basurajappa said we will have one session in refresher course, I said offline or online. He said offline. Then immediately I agree. If it is offline, I'll be very happy. But later onwards, I got to know that it is online mode. I was a little bit disappointed, but no matter. Anyhow, I'll be speaking, and at least I could see some of the faces. Given a chance, kindly switch on your videos so that at least I can see you guys, and then I can speak a little bit. Well, when he said about this refresher course, he asked me to take something which is related to all. I thought of entirely discussing on my research, but when he asked me, don't speak only research, better relate something general to all, then come back to your research. Then I said, okay, let me speak something which is interesting about my own area of research, where I am working in the last 2 decades. For this session, I hope I am audible and you can understand, right?
Yes, sir.
Yes, sir.
Well, for this session, I took a topic: gut microbes, no longer forgotten organ. It looks to be weird, actually. Whether it is an organ, can we call it as an organ? Because nowhere it was mentioned in our textbooks, actually. Nowhere it was told like gut microbes are the organ. But I also thought like that when I heard, but later onwards, when I dig into the details of gut microbes, then I thought, how can we forget this, and which are so important? Well, friends, we all taught during our primary education, including our high schools also. Animals are made up of with cells, including human beings. Our body is filled with different kinds of cells. The basic building blocks of our bodies, our tissues, our organs is mainly made up of with cells. And this was believed for so long time. I said it was believed for so long time. The reason was, recently, like till 1990s, people thought like that. But during the 1990s, there was a major buzz around the world. Like some of the major scientific platforms made a cover story saying that some catchy words like some examples or some cover stories what I am taking from the reputed scientific platforms like Nature or Scientific American. They said that your inner ecosystem in your body, microbes outnumber your own cells. Who is exactly controlling them? Then in the Nature, they said the body microbes outnumber our own cells by more than 10 times. One interesting a quote which was given by The Economist, which is a leading a publication. They said microbes makes the man. It was like a fascinating word. In continuation to that, at the center there is one cover page story which tells that from the Annals of Science, which tells that germs are us. I really like that picture actually, the cover story. A toddling kid is being attracted an ocean of microorganisms. You can see that actually. And it is being attracted and they may colonize in that. So how to believe this actually? Really, our body is made up of with microorganisms. How to justify this? How are these scientific platforms are telling that germs are us, germs make the way for our body, germs are working for our body, microorganisms are working for our activities. So that's what I am going to discuss. And I'll try in this 1 hour session. I'm planning to keep it for around 15 minutes from here onwards. And last 15 minutes, I love to have an interactive session, which we always like to have an interaction session whenever we were sitting in a refresher course. Instead of listening, discussing would be better. And I hope if somebody is working in the same line, you can also throw a light on this. And if you are really working in association with what I'm going to speak, your suggestions, your questions are most welcome. So with a small preamble, I am taking into my topic, microbes in our body. Well, the human microbiota, it is not recently which has come out. During 1600 to 1700, the father of microbiology, Antony van Leeuwenhoek, he has shown for the first time there is a microorganism, the discovery of microorganism to the whole world. Probably you all know the story behind that. He was a cloth merchant. He was very much interested in designing the clothes. He was expert in micro artist. He was giving different designs to cloth. While observing under magnified lenses, he could see some germs. In order to confirm that, he took the teeth crevices and observed under microscope. He took different materials in our body, observed the microorganisms in it. The whole world was excited. Yes, there is microorganisms which are tiny in nature, which which cannot be seen. We need microscope for that. We all scientists gave an importance to the microorganism, but somehow we forgotten where it was isolated actually, where it was seen actually. And that was from the mouth. That was from the human body. But this concept was forgotten for many days. In 1990s, they thought in this line. So let us look into it and how exactly our body is made up of with microorganisms. Like to tell, we have in our body an approximately 10 trillion human cells. But the number of microorganisms, what we have in our body is almost 10 times more than that. That is like 100 trillion bacterial cells in our body. Not only bacteria, other organisms also. So not just cells. And also, the number of genes what we have in our body, it is estimated like 22 to 23,000 genes are present in our body. But the 100 trillion bacterial cells estimated to have around 100 times of more than the human genes. Like 1:100. 22,000 genes, human genes, 20 million microbial genes. All are being expressed. All are being very active. When the number of genes are active, which are outnumber the number of my human genes, number of bacterial cells is almost around 10:1. So in that sense, whether the body is filled with cell or a microorganism. So when we say something like this, where are they present then? Whether all these 100 trillion bacterial cells are present only in one place? Are they spread throughout the body? Yes, of course. They have distributed in the different parts of the human body. Put together, we call it as human microbiota. To our surprise, wherever they are present, most of the time, they exert beneficial effects. Friends, many times we get infected actually. We get microbial diseases actually. That microbes come from outside or inside the body. One microorganism can cause a disease. Believe that there are 100 trillions of bacteria are there. If all are pathogenic, probably we wouldn't have imagined the life at all. And all those bacteria, where are they present? They are working for us. Now, I have few examples here. Our nose is known to have a microbial diversity. They are involved in the mucus production. They are involved in producing antimicrobial chemicals to fight against pathogens. In our mouth, we have microorganisms. They do assist for the digestion. And also they avoid many pathogens. They ward off pathogens. The next one, in our lungs, there is a microbial colonization which lubricate the pulmonary tissues. From lungs, next we have a stomach. In stomach, there is a huge microorganism in continuation to that colon. So maximum number of bacteria or microbes are present in the region of stomach and colon that I'll be discussing in my future slides. If they are present in the stomach, they are involved in preventing gastric complications. When they are present in the colon, most of the microorganisms which are there for the digestion of different carbohydrates, complex carbohydrates, undigestible matters. And they are also known to present in sexual organs. The sexual organs, the pH of the sexual organs is maintained by the microflora. And also they are known to produce some of the antimicrobial substances like H2O2 and other secondary metabolites to ward off the pathogens. And of course, the skin microflora. So the skin microflora being given more importance actually. There are microorganisms which are harboring on skin. There are certain examples which are maintaining cleanliness on our skin. Really, whenever we are using more of disinfectants like soaps, detergents, and many other very aggressive chemicals, we are going to clean the skin. Not only we are going to clean the skin, we're also going to kill the microorganisms, which works for us. So this is how they are associated. But among all, maybe from nose, maybe from mouth or lungs, I am planning to discuss more on human gut microbiota. The reason is the human gut microbiota recently a buzzword, a hotcake for research actually. And probably all of you might have heard gut microbes. And there are so many research which is taking place on gut microbes, engineering the gut microbes, personalizing the gut microbes. Gut microbes are involved in many other aspects. But when we think in this direction, people thought that, how do you say without microorganisms you can't live? And you say that your body is filled with microorganisms. You say that microorganisms are must for the living. Is it true? Do we need to believe this actually? So some of the scientists have carried out some experiments, and probably you all know that the experiment of notobiosis. Notobiosis means providing a clean environment, a sterile environment where there is no microorganisms. Like some of the model organisms have been developed, grown under completely sterile condition where there is no microorganisms involvement. So some of the model organisms, germ-free model organisms, which are grown in a aseptic condition, axonic condition, or a sterile condition, and those organisms are called notobiotic model organisms. There are several notobiotic model organisms being developed, like fish, plants, animals, guinea pig, like many many animals grown under notobiosis condition and checked the role of microorganisms in that. I think probably you can see this is a place where it is called Notobiosis lab. Everything is sterile here. They made sure that no extra environmental infrared. They cannot come inside here. No other microorganisms to enter here. But the results have shown that the animals, the plants, which are grown a microbial free, they got to know that very poor survival rate. Some of them have shown a very short lifespan. Some of them have shown abnormal behavior, abnormal growth, abnormal eating pattern. And when they have grown the plants have shown variations. Like they did not give the flowers. They did not grow completely. So in that sense, so they concluded that association of microorganisms are must to stay alive, to be active, to grow normal. So among all the microorganisms associated in our body, the largest amount of microorganism which is present in the colon, as I said. So when people started thinking about this until 1990, after 1990, people started looking into the colon region. How many types of bacteria are there? What kinds of microorganisms are there? Like whenever we discuss about gut microbiota, we come across with three terms. One is microbes, another one is microbiota, and the third one is microbiome. Like just to differentiate these three terms, microbes, you all know that they are tiny organisms like virus, bacteria, protist, algae, and fungi. But microbiota means a microbes living in and around the intestine is called microbiota, gut microbiota. Whenever we use the terminology microbiota, it refers to gut. And another terminology which we come across is microbiome. The microbiome refers to the genetic material present in all these microorganisms which forms microbiome. Please don't confuse with these terminologies. Whenever we say microbiome, it refers to a genes. Whenever we say microbiota, it refers to a gut microbiota. So gut microbiota is known to have, as on today, approximately more than 1,000 species of bacteria. And possibly over 7,000 different strains have been isolated and characterized. Friends, this 1,000 and 7,000 is not completed. We still have 80% which remains uncultured. Means we are unable to culture them. We are unable to grow them in our laboratory. So we are supposed to go for metagenomics concept. I think probably you might have discussed metagenomics in your previous discussion. So 80% is still like we don't know what they are. So when I say the colon, when we look into this colon and the stomach, small intestine, we have seen different diversities. Why there is a diversity in colon actually, GI tract? Why not a single organism is present? The reason is, friends, you all know very clearly, the GI tract is known to have three regions. One is stomach. The second one is small intestine. And the third one is large intestine, which is said to be colon. In all the three regions, we have a different pH conditions. Different pH of an habitat promotes different kinds of organisms. Like in stomach, it's an acid challenge, wherein we have an acidic condition of 1 to 4. In that, we could see the number of microorganisms around 10 to the power of 4 per gram of sample which is being analyzed. And the major ones are Lactobacilliaceae members, means the member of Lactobacillus. Probably you might have heard. Otherwise, I'll come back to that point in my future slides. And coming to the small intestine, there is a broad range of pHs there, around 5 to 7, 5.5 to 7. So the moment the pH increase takes place, which is nearer to the neutral, the increase in diversity has been observed. Increase in numbers are also being observed. From 10 to the power of 4 to 10 to the power of 7 to 10 to the power of 9 samples being up, 9 number of organisms being observed. But in the colon, where there is a neutral pH, most of the digestion, most of the major metabolic activity which takes place in the colon region, because the diversity of microorganism in the colon or the large intestine is huge. The numbers are also huge, like 10 to the power of 11 to 10 to the power of 12, which is almost 3 times more than in stomach. Like different types of organisms have been observed there. In colon also, there is a bacteria which is said to be Lactobacilliaceae, that is lactic acid bacteria. I am stressing this bacteria because my future slides will be having more on this. And other than Lactobacillus, there is an Enterobacteriaceae members, Enterococcaceae. And there are different microorganisms. The question is, what do they do? They are present from 10 to the power of 4 cells to 10 to the power of 11 cells. Do they have really any work to do over there? Or simply they are sitting? Whatever we are eating, they are also eating. Whatever we are digesting, they are also digesting. Will it be something like that? Friends, whatever I listed here, of course, only one is my cup of tea and rest are the research being carried out. But most of the functions are very much important in our body. The first and foremost important is breakdown of foods, especially indigestible and of course digestible food. Whenever we eat, We are not supposed to presume that only we are digesting the food. Along with we, microorganisms are also digesting the food. Why are they digesting the food? And they don't have just metabolism. They have a huge co-metabolism. One bacteria undergoes a metabolism, produces one product as an end product, and this end product is being used by one more group of organisms, and they work upon that end product, and they produces one more end product, are intermediate products, and these intermediate products and end products are being utilized by one more microorganism or a group of microorganisms, and this is what we say it as co-metabolism, which is very active. So whenever they are present in that, they supply energy to the gut. Actually, our gut needs a lot of energy. The major part of the energy comes through the metabolism from the microorganisms, especially the metabolites and the secondary metabolites which are being produced by the microorganisms. And not just energy, they are also known to make many vitamins like vitamin K, vitamin B12, folic acid, which are very useful for us. And whenever they are present in our colon or a gut, they have one wonderful mechanism. That mechanism is training immune system. Always our immune system gets triggered whenever there is a foreign body enters. Our foreign body, our body should be able to differentiate which one is a pathos or a pathogenic organism or which is a good organism. Because whenever we eat, we also eat many microorganisms, knowingly or unknowingly. I hope you all will agree with me actually. So when we eat pathogenic also, we eat good organisms also. Whenever we inhale the air, we take microorganisms too. Whenever we drink the water, we take microorganisms too. So the gut microbiota helps in differentiating which is the pathogen, which is not. And another important task which is being done, whenever we eat, we're also known to take some of the toxins, like endotoxins being produced by bacteria, exotoxins which are being produced by different microbes, mycotoxins which are being produced, fungi. And when these mycotoxins are toxins which enters in our body, they exert health complications. But these gut microorganisms have the ability to degrade or to transform those endotoxins and mycotoxins. And this is one of my research aspects, what I'm exactly working. I'm working on a lactobacillus, which is known to control mycotoxinic fungi, which is known to degrade mycotoxins. Of course, I'll show some of the slides in my future slides. And other than that, they also protect against pathogens. They don't allow to colonize them. Whenever we ingest, whenever we take a pathogenic microorganisms into our body, in order to exert the diseases, in order to make the disease condition, they are supposed to colonize on the body. But these organisms, they don't allow to sit and colonize on them. They produce antimicrobials so that they try to avoid it. And nowadays, probably you all know xenobiotics, which are man-made, which are not naturally present in the present environment. we have a major problem with this xenobiotics. And some of the research has shown that these gut microbiota have the ability to degrade these xenobiotics, have the ability to get rid of this problem. And another one, a gut-brain axis. Is there any relationship with microorganisms and our brain? A huge research is being carried out currently on this. And some of the research findings clearly tells that there is an interaction between microbes and the brain. Like the gut microbiota, which is very much involved in producing one of the metabolites, something like serotonin. Probably you know better than me. A serotonin, which makes your activities, your brain, your mood, and other aspects. So serotonin production is mainly, some of the serotonin production is also by the gut microbiota. And they're also involved in exchanging many components with gut and brain axis. So probably now you are thinking, are they simply working for our betterness? Don't they cause any problems to us at all? In that sense, then why are we getting diseases? Why are we having so many complications? Why are we having many syndromes? Friends, whenever any microorganisms favors for a single type or a pathos, whenever our body promotes for the growth of one particular kind, which is known to be a pathogenic, that leads to a disease, that leads to a complication. Then what exactly happening? Not just gut microbiota, we also have pathobionts in our body. Whenever there is a drastic reduction of immune system, when we lose our strength, the microbes which are present in our body starts its expression. Like Japanese scientists, they tell that all the natural deaths, so-called whatever we say, due to age or after a certain time we get natural death, that natural death is mainly due to the growth of these pathobionts which are present inside our body. When our body strength comes down, when the immune system is being compromised or comes down, automatically these pathobionts starts growing. So they become potentially harmful species. Like good examples are, we have candida in our body. So whenever there is an immune system decrease, then automatically it comes. And most of the activities what we are doing, the kind of food what we are encouraging nowadays, the fast food generation, our monotonous food, making something like dysbiosis, that is losing gut microbiota. So change in the population, change in the diversity is also leading the complications. And the obesity, and nowadays this has become a major lifestyle disease. Many are obese. And people have studied about this problem. Whether is there any relationship between obesity and the microorganism, gut microbiota? research has clearly shown that people whose intestines have smaller and less diverse bacterial populations, they are known to be obese. They have obesity problem. A study has been conducted. They have taken 2 mice. Same gut microbiota, one is being given, one was not. The mice which was not given any gut microbiota, that was obese. At the same time, the mice which was given with gut microbiota population, that was normal. Like similarly, many studies have been conducted in different model organisms. And other than this, they are also known to have implications like IBD, autism, like communicative social behavioral problems, and of course the diabetes. And still many more research findings are very much required to prove the role of gut microbiota in diabetes. There are so many publications which have come out, but not exactly, which tells that there is a huge role of microbiota in making diabetes, and of course the autism. Then in this situation, to avoid the encouragement of pathobionts or dysbiosis or harmful organisms growth, what are we supposed to do? And probably for many complications, the single sutra is, a remedy is, take a better food, take a balanced food. Friends, the food what we are eating is not only for us, we are also eating for microorganisms. We are also giving food for microorganisms. They are depending on us. If we eat street food today, they also eat street food. If we eat vegetables today, they also eat vegetables. If we eat less nutritious food, they also eat less nutritious food. So eating balanced food is one of the important things. And the second most important is taking probiotics. So I'll try to discuss around 5 minutes on this for probiotics, because that is my area of research. And the third one, which is recently being approved, eating our own shit. Shit, we can't imagine this actually. How can we eat our own shit actually? Friends, it is recently being approved by FDA, Food and Drug Administration. First fecal microbiota product, Rebiota, which is in the form of capsules. Of course, we are not eating shit, but the microorganisms which are present in the shit, in the excretion matter, they are extracting that microorganisms. They are providing the habitat or the nutrients in a capsule form. It is being advised to take as a fecal matter transplantation, FMT. The US Food and Drug Administration approved Rebiota, which is a first fecal microbiota product. The name is Rebiota. It is available in the market to treat against some of the complications. So coming to the probiotics, which is my part of research. Probiotics are a bacteria which are useful bacteria found in human gut, both in stomach and as well as colon. And it is defined as consuming live microorganisms which are living. When they are administered in an adequate amount in our body, an appropriate amount, we can't take this much, either we can't take this much. We are supposed to take exact amount. They exert or they give beneficial effects as per the WHO, World Health Organization. Probiotics considered are given GRAS status. GRAS, probably you all know, generally regarded as safe. You can use. But not all bacteria which are said to be probiotics can be directly used. They also need certain safety assessment. But for probiotics, the moment if you say probiotics, no need to have that safety assessment. All probiotics are considered to be safe. All probiotics have been given GRAS status. So hence we can consume throughout the world. So whenever we say probiotic, they are harmless, which can adhere to our mucus, which can tolerate to acid and bile, since they are present both in stomach and as well as colon. They can colonize on our epithelial cell. They have antibiotic susceptibility. They can stay alive in the acid condition. So knowing these conditions of probiotics have been used for the different purposes. A huge research is being done on probiotic, like to lower the blood cholesterol, to inhibit the pathogen, to produce vitamins, to prevent diarrhea. Especially nowadays, any kids, if they go and say that they have a diarrhea, the doctor will be prescribed one sachet having only lactobacillus and one yeast that is called Saccharomyces boulardii. A different compositions of probiotics, which is a treatment for diarrhea, which is available in any medical stores. And also to treat cancer, research is being proven to have more effect, and many other purposes. So in this direction, to collect the microorganisms of probiotics, I have screened several fermented foods which are known to have probiotics. Like I have collected freely occurring in the different regions of South India. And in my lab, from last 1 decade, we are involved in creating a probiotic library. And so far we have created a known of around 251 probiotic strains. This library we are using to prove as a probiotic organism from different foods, and also to identify which probiotic organism is this, and to have an additional effect such as antimicrobial activities and anti-mycotoxinic activity, and also mycotoxin detoxification studies. And this is what we are doing actually. Isolate a bacteria from fermented foods, prove them to be probiotic, and try to screen which one is having an additional benefit, like which can control different pathogenic organisms, which can control mycotoxinic fungi, which can degrade mycotoxins or detoxify mycotoxins. And we do conduct different tests in order to prove these objectives, like antifungal activities, antimicrobial activities, anti-mycotoxin studies, and to prove probiotics. Some of the slides which I am showing, which are recently being published, this work which is being published recently in the Journal of Food Bioscience, which is published from the elsewhere, having an impact factor of around 5 to 6. In this work, we were interested in knowing one of the probiotic organism, Lactobacillus plantarum, to inhibit the mycotoxigenic Aspergillus species, like different Aspergillus species, which are known to produce different mycotoxins like aflatoxin, acrotoxin. And we have screened their ability to control in different dimensions, whether it can control on a Petri plate, which can control in a broth, whether it can decrease the mycelial mat, and it has proven to have an inhibitory activity against the Aspergillus species. And not just it controls the mycelia, it is also delays or inhibit conidial germination, because conidia is one of the major or aggressive way of distribution of fungi. So the conidia being produced by Aspergillus and they are being released, and wherever they go, they germinate and becomes one more new colony. So in our study, we are interested in inhibiting conidia germination. It has shown the CFS, the metabolites which is being produced by the Lactobacillus plantarum, shown to inhibit the conidia. And we have also tried to see under electron microscope how exactly it is interacting with the fungi. We found that the whole hyphae is damaged, whole hyphae is being attacked by the bacteria, colonized by them, and makes deformed. And we have also tried to know they're useful in poultry feed and also the peanuts, because in poultry feed and peanuts, the problem of Aspergillus is more. Whether this can be used as biopreservative or not, it is being proven. It can be used. This is one more research being done by one of my research scholars. Here we have targeted Fusarium verticillitis, which is known to produce one more mycotoxin, that is fumonisin, from a different lactobacillus species, that is Lactobacillus brevis, which has shown its good activity against Fusarium verticillitis. Then not only Aspergillus acraceus, Aspergillus flavus, we have also tried with one more mycotoxinic fungi, that is Aspergillus parasiticus, which is known to be a potential aflatoxin producer. Especially the CFS, or the metabolites which is being produced by the Lactobacillus plantarum, has shown clear inhibition of mycotoxigenic fungi, and also which has shown the ability to degrade or to biotransform or to absorb the mycotoxin. So our lab is not just targeted on fungi. We are also interested in knowing the effect of these probiotic bacteria, which we have isolated, against different foodborne pathogens, especially bacteria. So in this work, this is also being published in one of the elsewhere journal, biotechnology reports. So in this, we have tried to know the impact of Lactobacillus plantarum against Salmonella. So we have seen a good antimicrobial activity against Salmonella, not just controlling the Salmonella growth, it has also shown the anti-biofilm forming ability. These Salmonella are one of the major biofilm producers. They can colonize as a biofilm. And the CFS obtained, or the organism what we have screened, has the ability to decrease or to inhibit the biofilm formation. Here you can see in the left bottom corner the electron microscopic images. And when we try to inoculate both the organism put together, we have found that the lactobacillus can reduce the growth of Salmonella when we have inoculated simultaneously. As a co-cultured method. And the CFS, which is known to have different metabolites, in this study, we got to know that the characterization of CFS has shown that some of the organic acids which are present in the CFS has shown to be effective against Salmonella. In the same line, we worked against one more foodborne pathogen, that is Listeria monocytogenes. In all the assays, whatever we have conducted, like reduction of growth, biofilm inhibition, antimicrobial activity of the CFS, shown to be efficient in controlling Listeria monocytogenes. And of course, this result is yet to be published. We are waiting to characterize the CFS, as we found the active component in CFS to be the protein or peptides. So we are involved and we are interested in characterizing that peptide. Will there be a bacteriocin, or is there any other type of peptides which are involved? So we have sent it for IIT Bombay for the characterization purpose. I just thought of discussing this also in this session. And we are not just interested in only probiotic bacteria, we are also interested in yeast probiotics, that is fungal probiotics. One of my research scholars who is working on yeast probiotics, who has isolated many yeast probiotics, and she started screening against different pathogens. It's a preliminary result what I'm sharing today. Parallelly, some of the most effective isolates among all the isolates what all we worked in the last 1 decade. One of my research collaborator from Canada, he was interested in sequencing. My student has visited his lab, and wherein we have completely sequenced 2 organisms, that is Lactobacillus brevis, Lactobacillus paracasei, 2 organisms which are having high antimycotoxigenic activities. The whole genome sequencing has clearly shown that some of the genes which are very specific to antimycotoxigenic in nature. And of course, whatever the data, what I'm showing, the functional categories, which are shown all the genes. But our gene of interest were also being observed, like T3PKS, that is type 3 polyketide synthase, and also other important NRPS, non-ribosomal peptide synthesis, which is being observed in both Levy's and as well as paracasei whole genome sequencing. One is being published in Frontiers in Microbiology, one of the leading journal in microbiology, having an impact factor of around 6 to 7. And one more is yet to be published, that is paracasei. We are just waiting to get some more results on this. And this is one of the preliminary results what I am discussing today. Our lab did not stop only looking into only in vitro studies. We also tried to know their effect in animal studies. We have used poultry as well as mice studies against mycotoxins And our study has clearly shown that the bacteria which is being given to the poultry known to have a beneficial effect against mycotoxins, which was clearly shown that I am not sharing that data because it takes again a too big aspect. And also in my previous discussion, I discussed about that. That's why I'm not discussing today. And recently we have done my studies also against one more mycotoxin, that is DON, deoxynivalenol, which is proven to be very effective. And the last slide, what I would like to show here is, whatever the bacteria we have given into the poultry or into the chicken, and probably you might be thinking that we have a huge number of bacteria in the gut. Why do you want to give one more bacteria? And how do you ensure that the bacteria, the one bacteria you have given is exerting the activity? So in order to prove that, we have tried to isolate from the colon of the poultry the same bacteria, whether we can isolate or not, using molecular approaches and of course the cultural aspects. And the first one is the control, wherein a small number of bacteria, lactobacillus on a MRS being isolated in the fecal matter. But in turn, the pre-colonized, a challenge study, and of course the probiotic given have shown to have more number of the bacteria what we have given. Since we have used the selective media, we could able to isolate only lactobacillus here. So in compared to the control group, we can isolate more number of bacteria. Then we pick the colony, we have characterized it, and we have again identified by using molecular aspects. And we proved that the bacteria, what we have given through orally or through feed or different mode of ingestion has shown to have a beneficial effect of the probiotic organism. So with this, friends, I would like to conclude my study or my discussion with a simple quote that many years we have been listening a one word called gut feeling actually. I was always thinking, whenever to make a strong decision, whenever to make very important decision, we say, my gut feeling says something like this. I really don't know what is exactly gut feeling actually. But after studying a little bit more about gut microbiota, I feel that gut feeling or a gut decision is not by you. It's a collective decision by you and as well as microorganisms. So we both are deciding as a gut feeling. So maintain a healthy gut, lead a healthy health. Now this is what my conclusion. And these are a few publications which so far what all I have discussed. I did not discuss much about my research. I just glanced it, and you can get most of the papers in my CV as well as in the university website, and you can download and you can get more information on that. So all these papers not from my work actually, it's a collective work. I have blessed with a wonderful research scholars who are working for me, and I would like to thank at this time to tell that we have worked something like this. And not just our own research scholars, and also the funding agencies. Somehow my research is being recognized by many funding agencies. Throughout my research career, I am having different research projects. Few are from UGC, one is from UGC, and few are from DST and SERB. And of course, foreign institution also, they are involved in helping or joining hands for my research work. So with this, I would like to thank you one and all for your patient listening. So I would like to have, I thought of winding up by 11 o'clock, and somehow it has taken me a few minutes extra. And I have another 10 minutes. I'm happy to interact with you.
Sir.
Yes, sir. Niranjan, sir. Thank you.
Yes, sir. First of all, we thank you for the very interesting and exciting presentation. One question, sir.
Yes, sir.
What determines the composition of this microbiota, sir, gut microbiota? Specifically, whether heredity has any role in that, I want to know.
Sir, this question is being asked everywhere, actually. Like, we don't get microbiota from our parents, actually. It is not like inheritance. They say that in an amniotic fluid, it's a sterile condition, actually, where there is no microorganisms. But some of the recent studies, they say that metagenomics approach, they found that microbiome, actually the genes in that amniotic fluid.
Yes, yes, yes.
So that genes might have entered into the a fetus or a baby, and they started expressing. And the first strike is the normal delivery. When the baby comes through the natural delivery system, that is the first encounter of microorganisms. That study is also being done. The C-section babies and normal delivery babies, actually, they found more healthy than the C-section. But the moment it comes out, all environmental factors influences the gut microbiota. And the kind of food also influences. And of course, I don't deny that it is also could be heredity, because some of the complications, what we have seen, which are related to colon, are also being inherited. So I would say maybe there is a role.
Yes, sir. Thank you, sir. Sir, one more thing. Suppose
Yes, sir.
if a person or a individual is exposed to some pets or animals from a very young age,
Yes, sir.
will this have any effect on the microbiome, sir?
Really, it's good for us, actually. We get many microorganisms from the environment, actually. That's why that Scientific America with the cover page has shown one kid is, a dog is licking, actually.
Okay.
It is providing good microbes, because whenever we are associated with dogs, we don't fall sick, actually. Many times we get good bacteria. But of course, sometimes when we take the hairs and other matters, that becomes a complicated, actually.
Thank you. Thank you.
Thank you. Thank you, Niranjan, sir.
Sir, very good morning, sir.
Very good morning.
Good afternoon.
Sir, I am Dr. Kalishwaran from Tamil Nadu AVM Sri Pushpam College, sir.
Yes, sir. Yes, sir. Please.
Department of Zoology.
Yes, sir.
Sir, I'm working in the same discipline of probiotics in the help of for immunostimulation activity in fish.
Okay. Okay.
So, in which I had two questions there in my mind. So, one is most of the research works and the papers, we can see that fish immunology, because it is in the water itself.
Yeah.
So, we are doing that.
Yeah. Yeah.
And I met one person in the industrialist. He told that the probiotics and so many people are working in aquatic regions,
Okay.
but it won't work out in 100%. So what is your suggestion, sir? It is any justification is there?
I would say partially is right. The reason is sometimes one bacteria we cannot consider as a universal bacteria, which can colonize all the systems, actually.
Okay.
The place of habitat or the isolation habitat plays also crucial role. So if you are really interested in isolating a probiotic from fish habitat, then test for its efficacy. It is known to work better than a bacteria isolated from my colon.
All right.
So it is better to take a native habitat rather than different habitats. If you are really interested in working with plants, better isolate a plant organism and then test against it. Don't isolate from an extraterrestrial environment and then test for plants. There is no correlation.
Okay.
Yes, sir.
Okay. In which that in the colon cell we can collect it from the feces.
Yes, sir.
Feces from birds or anybody, anything.
100%, sir.
It is also mixed with some of the fluid substances.
Yes, sir. Yes, sir.
So pH is maybe play a major role for it.
Yes, sir.
So in which water pH and this feces pH, it may be more or less similar.
Okay, okay.
Whether I can utilize that particular bacteria to treat the fish disease or immunostimulate activities, whether it is okay, sir.
Sir, to say exactly, like whatever the fish intestine region or digestive region has a pH, better to take that organism. The moment you give, it has to colonize first fish actually.
Yeah.
So that's what we do whenever we isolate bacteria, whether it colonizes or whether it is tolerate to acid of a stomach actually.
Yeah.
That's what we do, bile salt and as well as acid tolerant.
Yeah,
So
sir.
you are supposed to do that actually. Then you can use that.
Okay. If it is accumulates, it is better, right?
Yes, yes. If it is if won't, then there is a problem actually.
Okay, okay. So do you have an idea? Just I am asking you that if I want to take the probiotics mixed with the materials, whatever we are given for diet purposes to the fish.
Okay.
So it won't disseminate into the water conditions.
Yes.
So what is your suggestion, sir? How can I give?
Sir, better to mix with feeds and make it as a composition in the feed. Because nowadays biofeeds are available. I am also working on biofeed actually.
Okay, okay, sir.
I am working on poultry. So I also came across with aqua feeds.
Okay.
Which are more contaminated with mycotoxins and other aspects also.
Obviously.
So
Yes,
better to mix it with feeds and then give it to them. So that they can colonize.
Okay, sir. Okay, sir. Sir, nice, sir. Thank you. So after this meeting, sir, I will contact you personally and then I will discuss.
You are most welcome.
Thank you. Thank you, sir.
Good afternoon, sir.